Welcome to Recovering Evangelicals, a podcast for people who were once very comfortable in the Christian faith until the 21st century intruded and made it very hard to keep on believing. I'm your host, Luke Jansen. Scott is taking a break today. Last week we talked to Dr. Michael Behe, a biochemist at Lehigh University and senior fellow at the Discovery Institute, about his perspective on intelligent design. He's a very vocal advocate of that position and a leading proponent of it, and has been for decades. When he speaks and writes about intelligent design, he often refers to the bacterial flagellum. In fact, the flagellum has almost become his signature, his mascot. However, Dr. Behe hasn't actually worked directly with the flagellum, and many of his statements about the flagellum made to support his ideas about intelligent design have been challenged by scientists, including those who actually do work with the flagellum. So to help us make sense of this, we reached out to about a dozen scientists who have done extensive work on the flagellum, coming at it from all kinds of angles and using a variety of cutting-edge experimental techniques. Today we'll hear from one of them. Dr. Nicholas Matsky obtained his PhD at the University of California, Berkeley, in the Department of Integrative Biology, then was a fellow at the National Institute of Mathematical and Biological Synthesis, based at the University of Tennessee, then began early career research at the Australian National University in their Division of Ecology, Evolution, and Genetics, and is now senior lecturer in the Department of Biological Sciences at the University of Auckland, New Zealand.
Yeah, thanks very much.
Nick, you've clearly had a long-standing interest in biology and evolution and spent years learning how to bring genetics and mathematics and statistics and various forms of logic and even geography, I understand, to trace through the evolutionary history of the bacterial flagellum. Tell me how you brought so many different disciplines to that one study.
Sure. Well, I guess you have to say my original interest was evolution in general. And I did really decide to go hard in that direction while doing, ironically, a degree in geography, of all things. Back in the day, I did a master's degree in geography. And it turned out to not really be my passion. And I did kind of realize around then that evolution was my passion. And one of the early subjects, originally as a hobby that I got involved in, was debating creationists, debating the intelligent design proponents. And one of their favorite topics, even 20 years ago, was the bacterial flagellum. And that was one that I realized, there actually hadn't been a lot of evolutionary work on it at the time. And so I got into it originally as just kind of an internet argument, but it really developed into this whole area that's worth studying. And now, I went and got a PhD in evolution, did phylogenetics.
Phylogenetics is the study of the origin of families of organisms, kind of like a genealogy for all living things or a tree of life.
Did phylogenetics. I did phylogenetics on biogeography, how species move around. But then I was lucky enough through some collaborations, to get back specifically onto the flagellum topic. And so now that's one of my research interests officially, with people in my lab and with labs that I work with, trying to further advance our understanding of the evolution of the bacterial flagellum.
Okay. Now, I think a lot of people who are interested in this story of the flagellum, many of them might think they might have this mistaken idea that it's a thing. I say that in air quotes, and what I mean is that it's one thing, that any cell that has a flagellum, it all looks like one image that they have in their mind. But instead, I understand there's a whole family of these machines called the flagellum, different types and subtypes of the flagella.
Yeah, so that's very true. I mean, most people, if they've heard anything about it, and we should review what the picture is that people have, but most people are thinking of the textbook illustration of the flagellum. So in a biochemistry textbook or an introductory biology textbook, they will, there will be a chapter on cells and the different parts of cells. And one of the famous parts of a bacterial cell is the bacterial flagellum, which is essentially, it's like a tail or a propeller. It sticks out of the cell. It's just a long filament that curls around. It sticks out of the cell and spins. And either one of those or several of those spin and they help the cell swim around. But the text, an introductory textbook is just trying to tell students that there's this thing called a flagellum. It's a Latin word for whip. Mm-hmm. Like many, many terms in biology, scientists look through a microscope or they look through another imaging system, and they just come up with a name for whatever they're looking at. And it was pretty natural to see the spinny thing and say, oh, that's sort of like a whip. We'll call it a flagellum. And so in a textbook, the first thing you want to convey to a student is there's a thing called the flagellum that helps the cell swim. And then as science advanced, a lot was discovered about the detailed structure and function of that system. But almost all of that work has been done on really one bacterium, which is E. coli, right? Escherichia coli, which is the sort of lab rat of bacteria that many, many people do experiments on. There are a couple other closely related ones. Salmonella, everyone's heard of salmonella because of food poisoning. It's a very, very close relative of E. coli. And there are a few others that people have done experimental work on, but that's sort of the paradigmatic or the textbook system. And so when people talk about the flagellum, usually what they're talking about is that textbook one. And you can learn a lot by studying that textbook one, but it is true that, just like there are thousands and thousands of known described bacterial species, and there are presumably millions and millions and billions of undescribed ones. Similarly, for flagella, there are thousands and thousands of variations that are known, and there will be millions and billions that are out there that are unknown. Now, you can overdo that point because everything that we call the bacterial flagellum, all of them share a bunch of things, and that is worth pointing out if we get to that point. There's the commonalities of that system are very interesting because there is a set of shared components that are pretty universal across those systems. But beyond that, there's a huge amount of variation and pretty bigger variation than people thought in terms of how the thing looks from the outside, in terms of the size and structure. So yeah, that's a lot of that's only recently been discovered due to new technology.
Okay, and in addition to the fact that there are these commonalities, you refer to them having very many of the same proteins, although different gene sequences underlying those proteins. But some of them, in addition to those commonalities, there are some very marked differences. Some of these bacterial flagella are going to be missing certain parts, and they still work with these.
If you were to take I and Mark Fallon did a study quite a while ago where if you take the kind of textbook list from E. coli, and it's got, probably say around 50 proteins, 50 different proteins, proteins, right, are encoded by genes in the DNA. So there's about 50 different genes in this kind of textbook system. And if you ask how many of those are found across all of the bacterial flagella, that's a pretty coherent research question that you can ask. And the answer is about 20 of them are pretty universal, maybe 23 or 4, depending. And so a bunch of the proteins in the sort of E. coli textbook system are actually not essential because they're not found in many other species that have a functioning flagellum. And even in that list of parts that really are pretty much universal as far as we can tell, some of them don't actually seem to be essential to function. They're probably helpful, and that's why they're always there. But, people have done experiments in some cases and knocked out those genes and still gotten a functioning flagellum. And so some of the parts are probably helpful, but not essential. But there is still a core there of probably 20 proteins that are, as far as we know, experimentally are probably essential to the function of the known flagella. So, that I think the, Michael Behe's irreducible complexity argument, in that sense, it has legs even after you do that kind of analysis, because there is still this list of proteins that seem to all be required to produce this very complex function of producing swimming ability. And, and so it's a good evolutionary puzzle. How did that complex of proteins come together? Presumably, if you think they're all essential, you couldn't gradually build up to 20 proteins if you need all 20 to produce the function. That's kind of the argument. And as a starting point argument, it's a reasonably clever one.
Now, we're not going to talk about this today. We'll do this next week with yourself and a couple of your colleagues, but we want to talk then about the story that, no, the flagellum didn't gradually build up through, let's just say for argument's sake, through 20 steps, and it was non-functional until it reached step number 20. But rather, we're going to talk about a whole sequence of changes from one type of an enzyme to a completely different, functionally different enzyme, and then another functional enzyme that's quite different, and a series of these steps through gradual changes that give rise to different enzymes, different structural components, different motor proteins, and in the end, through this gradual evolution of different kinds of things, one of the outcomes was the flagellum.
And I would say, if you wanted to bullet point it, you can say that by linking together several simpler systems with different functions, that's how you seem to get to a really complex system like the flagellum.
Right.
So you have these sort of subsystems that can do their own thing and are known to do their own thing out there in the world, and then linking those together in a few steps probably produce the basic ancestral flagellum.
Right. But the one thing that I did want to clarify, though, because I have been digging into this intelligent design, I'm trying to make sense of it. And the one comment that's often made, specifically with respect to the flagellum, is you can't mutate it in any way. You can't take away any of the parts, or it loses its function and the cell dies. End of story. And the fact that you've now told me that there's thousands of different kinds of flagella, different sequences, they've mutated through all these different ranges of sequences, and some of these are now missing parts, and they still work. So it really, for me, casts a lot of doubt on their claim.
Yeah, well, so this is a bit of the a bit of the moving goalposts part of the argument, right? There's been a history in this debate, especially when it was hot, in the 2010s, where an intelligent design proponent would kind of set up a challenge. They'd say, "Oh, this," and I actually tracked this once, where I asked how many parts are being claimed by the intelligent design proponents as being essential. Because I think, as I recall, it's been a while since I looked at this, but I think I was able to find quotes where certain intelligent design proponents would say the bacterial flagellum has 50 proteins and they're all required. And later on, it was another year or two later, they learned a little bit more. It's like, 40 proteins and they're all required. And then it was 30 proteins and they're all required. And it got down around somewhere like 20 and they're all required. And so you had this kind of, reducing argument a little bit, which is ironic.
Right.
Argument's irreducible complexity. Now, like I said, you pretty much can say that there's something like 20 proteins that are all required, as far as we know, in the systems we've looked at and experimentally. And so, you've still got the argument there, honestly, but it is reduced. And it's also true to say that once you fully appreciate the diversity of the systems and the diversity, each of the genes producing each of the proteins, each of those genes is a member of a gene family. And so you could pick any flagellum protein, and these days you can search it on a database of many other genome sequences, right? And you can take a gene and you will see that relatives of that gene, thousands and thousands of relatives of that gene exist, and none of them are identical in sequence. And you can put all of those genes into a big evolutionary tree, and you can ask which parts of those genes are highly variable and probably not very essential to function, which parts are highly conserved and are probably pretty important for function. For some of the proteins, the sequences can change so much that you can't detect any similarity, but nevertheless, the function is retained. And so there are cases where the sequences change so much, you've kind of lost the signal of similarity, but the function's still there. And you ask, how can you tell that? Sometimes you can have a chain of sequences that can link two very divergent sequences together, or sometimes the protein structures will be available. There are ways to, every gene in DNA makes a protein made of amino acids. It's a chain of amino acids, but they all fold up into some complex microscopic three-dimensional structure. And those three-dimensional structures can now be imaged in various ways. And you can sometimes, if you've done that for a couple different bacteria, you can compare those structures on the computer. And sometimes you'll say, those two proteins have almost identical structures, even though the sequences have diverged so much that there's hardly any statistical similarity in the sequences. And so, if you start counting how much variation is allowed in a particular sequence, right, you're a long ways from the position where there's only one functional sequence that has to be exact.
Right.
It's more like there are billions and trillions and bajillions of sequences that are functional. And that's not the end of the argument either, but that is a point worth making, right? It's a starting point.
Sure. Now let's talk a bit about how that rotary engine works. How does the bacterium rotate this flagellum? It takes energy to do that. Where does it get the energy to do that? And how does it convert that energy into a rotational movement?
Right. So this is the really stunning thing about this system, right? That blows people's students, blows their minds away, or the general public. There are now, if you go on YouTube, there are animations. You can't actually see the system literally in a microscope, right? It's so small. It's below the resolution of what you can see in a microscope, pretty much. The few videos you see where you sort of see what might be a flagellum, bacterial flagellum, are really bundles of flagella. Some bacteria stick a bunch of flagella out from different parts of their body, and then as they rotate, they're so long they kind of tangle up with each other, and you can sort of see that on a light microscope. But yeah, the actual system is so small that it's below, the wavelength of light that you could see with a light microscope. So most of what you see are cool animations, and those give you a pretty good idea. But anyway, if we're on an audio thing, so you got to imagine kind of a round cell, or maybe a oval-shaped cell. Then imagine tiny little hairs sticking out of it. They're very, very long, 20, 50 times longer than the cell. Then imagine those tiny little hairs are in kind of a spiral shape, and the individual, hair is rotating right down at the base. And so the whole filament beyond that is spinning. And then imagine, if you can imagine it spinning, 50,000 times per minute, something like that. Sometimes it's 20,000, sometimes it's 100,000, depending on the species.
Wow.
But it's very, very fast. Now that, to our human senses, that seems very fast. In molecular world, you're down at the level of atoms and molecules. Lots of things are very fast at that world, so maybe it's less surprising, but it's spinning thousands and thousands of times. So it's going very, very fast. Now, so that long, long filament is really mostly made up of just 10, 20, 30,000 copies of one single protein, just repeated in kind of a helical spiral pattern. And so 99% of the mass of the bacterial flagellum is just this one protein called flagellum. And so that's making that hair. But a whole bunch of other proteins are cooperating down at the base of that system to cause the rotation to happen. And so it's kind of funny when you talk about, how many genes and proteins are involved. There are tens of thousands of copies of proteins that are in the flagellum, but all of those proteins come from just about 20 different genes that make 20 types of protein, right? And so the most common type of protein is the flagellum protein that makes the hair. That's 99% of the mass. But these other 19 or so proteins are setting up that whole system and then making it rotate. So we could talk about how that works briefly. At the edge of the cell, if you imagine that oval cell, if you zoom in on it, there are typically two membranes. There's an outer membrane and an inner membrane. And to make this system work, proteins are manufactured inside of the cell. The DNA is copied to messenger RNA, and then ribosomes translate the messenger RNA into a chain of amino acids that makes a protein. That all happens inside the cell. So you have to get those proteins outside of the cell somehow. And so this is actually a common problem that bacteria solve. And they have a variety of transport or secretion systems that can move proteins across a membrane. And the basic way you do that is a cell has to form a pore, a hole in the membrane of some kind. And then it has to have some kind of system to control what goes out of that pore. And in the case of the flagellum, at the base of that whole system is something that we call a type 3 secretion system. And that's about 10 of the proteins that cooperate to form a hole in the inner membrane, and then they form a hole in the outer membrane. And then there's several proteins, 6 or 7 proteins that cooperate to form kind of a gate at the base that pulls out, especially the flagellum protein after it's been manufactured, and kind of squeezes it through the hole and squeezes it through the outer hole, at which point it sticks onto the outside and forms a tube. And then again and again, more flagellum proteins are squeezed out through that hole, and they go all the way through the end of the tube, and then they stick on the end. So this tube is grown tip outwards, right? So the protein subunits go through the secretion system, across the membranes, into the tube, and they get squeezed out of the end of the tube and get added on. And so just the assembly of the system is mind-boggling when you think about it. And yeah, there's more to say, but that's the starting point, yeah.
So basically, you've got this mini machine that squirts out proteins at the base of the bigger machine, which is the flagellum that rotates. Yeah. So a machine built on a machine.
Exactly right.
And then the rotation, basically, I understand that flagellum is kind of inserted into, let's call it a ball, which then rotates inside a donut. You've got this ball sitting inside a donut, and it's just spinning inside the donut. And that's how it works. How does that ball in the donut interact to produce this rotational energy?
Right. So yeah. So the ball, or the you might almost call the sort of the inner circular system, right? The secretion system. The proteins are being secreted through the middle of it, but that whole secretion system is spinning inside of the membrane. And the membrane's not spinning. The system is spinning inside a ring of proteins that are actually driving that rotation. And so those proteins are known as MOT-A and MOT-B, MOT for motor. So there's these two motor proteins, and they are what take energy and turn it into motion and push the system around in a circle to spin it. And so MOT-AB are a well-known part of the system. And the power source they're using is in the textbook system, not at all systems, but in the textbook system, it's protons. So H plus hydrogen ions, right?
Acid.
Acid, yeah. So bacteria, one of their ways of storing and using energy is to pump ions in or out to create a gradient so that you've got a higher concentration on one side of the membrane than the other. And that power storage is used to then power a variety of different reactions, including the generation of ATP, the energy molecule of the cell. And so in all of our mitochondria, in our cells as humans and all other animals, there's a hydrogen ion gradient that powers a machine that produces the ATP molecule that's kind of the universal energy currency of the cell. But that same proton, this is called proton motive force or proton ion gradient, it gets used for a variety of other kinds of reactions like pumping molecules in or out of a membrane, or in the case of the bacterial flagellum, gets used to produce that rotary motion. And so I forget.
And Nick, if I can,
yeah.
If I can interrupt for a second, for the listeners, these are going to be big words.
Right. Yeah.
A proton motive force, but I think it's helpful for them to realize that this is not really different from a battery. A battery uses electrons. This thing is a battery that uses protons.
Yeah.
The movement of these protons is just like the movement of the electrons in a battery. So essentially, you've got an electric battery coupled to this ball and donut spinning wheel, which is then connected to a hair, which is the flagellum.
Yep, that's the good way to think about it. Things like photosynthesis or other kinds of energy generation in the cell, right? Part of what they're doing is moving protons and electrons around to build up these energy gradients to store energy. And it's in your mitochondria, those are the energy factories of your cells. Yeah, they're using that sort of process. So that was kind of known for a long time, but then it was truly wild to discover that the way the bacteria are swimming is using that kind of power source as well.
Right.
It's truly wild. And I can totally see why creationists and intelligent design people would latch onto that system. It is incredible. And you're not doing biology correctly if you don't step back and be like, holy moly, that's amazing. Right.
Right. Yeah. Okay, so Nick, we were talking about this type 3 secretion system squirting out these proteins that then make the flagellum. And I want to really emphasize to the listeners this concept of self-assembly. You've got these proteins, these machines, cellular machines that are making themselves. And I want to explore that concept of self-assembly in a bit more detail because a lot of people will get an image in their mind when they hear scientists refer to proteins as machines. They hear the word machine, they start to think of mechanic or an engineer or a designer because that's just the way we understand cars and other machines. They all need mechanics and engineers and designers. So they start to think, okay, who is behind all of this cell biology? Let's talk about the self-assembly of the flagellum. Scott and I have already talked to our listeners about how proteins assemble themselves spontaneously. We talked about electrostatic and hydrophilic forces. We don't need to repeat that too much, but I understand even in the order in which these proteins are squirted out, that already begins to determine how these things self-assemble. Can you tell us a little bit about that?
Yeah, so there's two or so levels to that question, right? And there's a DNA sequence that gets copied to an RNA sequence that's then translated into an amino acid sequence. And, don't sweat it too much, the listeners, if even that is mysterious, right? But that's what we teach students about in kind of intro biology. But the thing to understand about amino acids, right, is that they have a three-dimensional structure. So there are a series of atoms that are bonded together, and they have a three-dimensional structure. And when they're linked together in a chain, they don't just float around as kind of one long chain. Because different amino acids have different sizes, Some of them are negatively charged, some are positively charged, some are hydrophobic, which means they're sort of repelled by water molecules. Hydrophilic is attracted to water molecules. And so as a result, they can achieve a more stable energetic configuration by lumping up. And so this chain folds up into some quite specific three-dimensional structure. It's not even microscopic, it's sub-microscopic, right? It's smaller than you can see with a microscope. But each protein, I'm oversimplifying, there are some proteins that don't have a stable fold, but many proteins have kind of a stable folding shape. And that shape can be discovered through various kinds of experiments. And it was a real key to 20th century biology when it was realized that each of these different proteins, some of them are enzymes, some of them are structural proteins, they take a specific three-dimensional shape, and that three-dimensional shape determines what other proteins they will stick to, what other molecules they will interact with. And so if you have a protein that breaks down a certain toxin, it will have a shape that specifically matches some part of that toxin and can help break a bond in that toxin molecule, for example. Proteins can be shaped to fit each other. And so many of the systems in the cell are a collection of proteins that cooperate by having shapes that match. And you can do it in the computer these days. If you have a bunch of protein structures, you can put them together in the computer and see how they all fit together to produce some larger multi-protein system. And so the flagellum is just one of the more spectacular cases of that. So once we've got that idea that we've got all these proteins and they have different shapes, the flagellum has 20 or so core proteins with different shapes. And I mentioned that there's kind of a pore or a hole in the inner membrane, and then stuff gets transported out to form the hair or the flagellar filament at the end. Between that, there's actually a series of proteins that cross the cell wall and they cross the outer membrane. And then inside of that, at the base of the flagellum filament, there are actually a series of, you'd have to call them adapter proteins or linker proteins. There's the tube that makes up the flagellum filament continues right down into the base of the membrane, right down to that bottom pore, the inner membrane. And there's about 5 or 6 or 7 proteins that each get to be usually a little bit smaller and smaller. And it's basically a way of taking that ring system and changing its size to kind of latch right in tightly to that inner core protein. And they're given names of, let's see, there's flag B, flag C, flag B, flag E, flag F. And they're all actually pretty similar to each other in certain core components, but they change size and they help basically bridge that gap from the inner pore out to the outer filament. There's also a protein called the hook protein that's at the base of the filament on the outside of the flagellum. And that's just a particularly bendy part of the filament. And having that bending ability helps the flagellar filament get oriented in a certain way. So it turns out those proteins, they're not just secreted at random, they're secreted in a particular order so that the inner ones are secreted first and the outer ones are secreted later. And then finally, once the whole system's been built up, the last thing is just turning on the flagellin protein, the 99% of the mass of the flagellum. Once the whole system has been built and the inner tube has been built and the hook has been built, then the bacterium just says, all right, the rest of it's just going to be flagellar filament, and it just secretes that tens of thousands of times. So, but it's been this massive, 30 or 40-year effort, 50-year effort to figure all of that out via a lot of amazingly clever experiments, figuring out how that works. And it's only been done, it's only really been done in detail for a couple of bacteria, usually E. coli and Salmonella are two of the main ones. But it looks like it probably works in a pretty similar way for most bacteria, although there are known variants in some of those proteins.
Right.
And there's one other piece of it, right, is that proteins fold in part because of their amino acid sequence, but in part because of the local environment they're in. And in the case of the flagellum, the local environment, when the flagellum is being built, right, is sort of that specific environment of being squashed up in the tube and then popping out. And so when that happens, the protein is in kind of an elongated shape when it's going through the tube. And as it squeezes out at the end, it takes just the right shape to latch in to this building assembly of filament proteins. So yeah, it's all quite amazing. But yeah, self-assembly of proteins is the key concept. And you kind of got to imagine it's a little bit like Lego blocks or puzzle pieces, right? If you imagine you were able to take puzzle pieces and throw them in a box, and then jiggle it around as such that sometimes the right shapes matched each other and stuck together. That's a little bit what like proteins are doing,
Right.
except they're doing it in 3 dimensions in a sub-microscopic.
As it turns out, Nick, we actually, when Scott and I talked about this, we used the analogy of a kitchen drawer full of spoons, spoons that had been slightly magnetized with a north and a south pole.
Oh, yeah.
And when you jiggle this drawer full of spoons, they'll begin to self-assemble. Spoons will stack on top of spoons, north pole to south pole, that sort of thing. And it's just a self-assembly.
Yeah, so that's quite amazing by itself. But yeah, it can take a while. It can take students a while to get their heads around that because all of this, you can sort of have these analogies at the human scale, but once you really understand what's going on at the molecular world, right? It's it's sort of like the movie Ant-Man, right? Everything works different down tiny scale. And so
right.
Everything is jostling, everything's moving at that scale, and everything's constantly jostling. And so, but yeah, things like the charge of atoms, right? Positive charges attract the negative charges, and the shapes of the physical shapes of the atoms create these 3-dimensional patterns. In some cases, you get almost a lock and key kind of system. The
right.
proteins, lock and key stick right to each other, and once they're stuck together, they will either stick there really permanently, or at least they will statistically stick together more commonly than not, and thereby self-organize into a bigger system.
Okay, so that's one aspect of protein biology that I wanted to demystify for the listeners, that whole idea of self-assembly. The cells don't need a little person inside with a hammer and a screwdriver putting these things together. The proteins assemble themselves. But there's another aspect to protein biology that I wanted to demystify, and that's basically how proteins work. We've already talked about how it's the shape of the protein that defines its function. And I think the listeners, when they hear about a bacterial machine like the flagellum, they think, okay, car engine has got pistons inside cylinders and a fan belt, connecting gears interacting. So they picture all these moving parts in a cell. The proteins do not work in that way at all. Basically, it's a shape shift. The proteins just change their shape, and that produces the function. So an ion pump molecule bends in one direction, and it sucks up an ion. It bends in the opposite direction, and it pushes an ion out. An enzyme molecule bends one way, and it pulls two halves of a substrate molecule apart, or it bends the other way, and it clicks those two halves together. And the rotary engine molecule bends one way, and it takes a step to the right, or it bends the other way, and it takes a step to the left. So it's just simply a coordinated series of shape shifts of the protein parts. Is that?
Yeah, that's
what do you say to that?
Yeah, that's pretty good. If you get into the details, right, it all becomes kind of this noisy, sub-microscopic, noisy process because everything's moving all the time. So every protein that you think about it having a kind of a central structure, but really, and these days they simulate this on the computer. All these proteins are wiggling back and forth all the time in kind of every direction. And so whatever the classic quote unquote structure of a protein is kind of its average configuration. And then, but yeah, many protein functions occur when the average configuration shifts to a different average configuration. And that may, put force on another molecule, breaking it apart. That may push, that may be one protein pushing on another one and moving it. And that may be, helping move that rotary base of the flagellum, moving it in a circular direction. That's how it works. And often those moves are energized by some other power source, one of which can be the flow of positively charged hydrogen ions, like we talked about. So yeah, there's
okay.
Yeah.
So Nick, I do want to introduce one more bacterial machine. We've talked about a few of them now, but I want to introduce another one because we're going to talk about that next week. And this machine is basically, it's another kind of a pump, not one that pumps ions. Nick talked about some certain bacterial machines that move ions in one direction or the other. This one makes energy molecules, the gasoline for almost everything that cells do. And this is a pump or an enzyme called ATP synthase. So, Nick, can you tell us a little bit about this enzyme?
Yeah, so ATP synthase, it's another molecular machine. And by molecular machine, we typically mean, in some sense, any individual protein is a machine, but typically we mean a collection of different proteins encoded by different genes, where those proteins assemble into some kind of unit that then performs the function. And so, unlike the flagellum, which is 20 plus proteins cooperating. The ATP synthase is more like, I'm going to say 8 or so proteins cooperating. It varies a bit depending on the system. Again, most of what's known about it comes from a couple of bacteria, one of which is E. coli. And so again, in the textbook, you'll see typically the E. coli system, or actually in your mitochondria, most of our cells in humans and other animals contain these little energy factories called mitochondria, which are really themselves just very, very reduced, simplified bacteria. And there's a whole story there about where that came from. But yeah, inside each of our mitochondria, there are thousands and thousands of copies of these ATPase machines. And what they do, ATP is adenosine triphosphate. And that triphosphate is the energy storage part of the molecule. And by breaking off one phosphate group, a bunch of energy is released. And so we've got in the ATP synthase, in the kind of classic system, it's got 8 proteins, but they're really characterized into 2 groups. And one is the FO subunit and one is the F1 subunit. And the FO subunit is in the inner membrane, and the F1 subunit sticks out below the inner membrane into the inside of the cell. And that F1 subunit is the part that's interacting with ATP molecules. And depending on the specific system, it's either generating ATP or it's burning ATP. Whereas the FO subunit is the one conducting protons, again, typically, not quite always, but usually the proton gradient is being used to power the system. And by linking those two together, you link the flow of protons to the generation of ATP. And so it's a way of converting energy between a proton gradient and a chemical energy stored in the form of ATP.
So to put an image in the minds of the listeners, then tell me if this is an accurate representation of what you just said. Basically, if they can imagine a turnstile, like the kind that you have in the subway or in a department store. People walk through this turnstile that spins around and just moves people in one direction. So now you've got 2 turnstiles, one that's working with the protons, moving through the turnstile in a certain direction, and the other one that's moving the 2 halves of ATP and clicking them together to produce ATP. These 2 turnstiles are connected. There's a gear, kind of like a yo-yo. If the listeners can imagine a yo-yo, it's got 2 halves, and if you turn the one half of the yo-yo, you're going to turn the other half. They're connected. And this is how that ATP synthase moves protons or uses the movement of protons to click together the 2 halves of ATP together. Is that an accurate picture for that?
Yeah, that's accurate. And and so that's another amazing system, right? By linking 2 rotary sub pieces together, and especially there's these stalk, we call them the external stalk, right? There's these long, skinny proteins on the outside to kind of keep them from rotating freely. If you knock out those stalk proteins, right, the system, things rotate, but energy isn't transferred because there's nothing keeping them from just freely turning. You want the turning of one system to change the shape in the second system, thereby changing the shape of the proteins, thereby making them, for example, click together the pieces of the ATP molecule, right? So yeah, it's quite a spectacular system, and there's a number of rotary machines known in bacteria.
Now, there's one other thing that I want to say about this that I think will become really relevant next week. Again, I use the image of this yo-yo, the 2 halves of the yo-yo, one of which is working with the protons, moving the protons. The protons are spinning the bottom half, which is connected to the top half and does work over there. But we have the same thing, Nick, I believe, at the flagellum and in the type 3 secretion system. You've got the bottom half of the yo-yo being moved by protons or sometimes sodium ions, and then that bottom half is connected to the flagellum. So it's the movement of those protons through that yo-yo half that's spinning the flagellum. Is that fair?
Yeah, that the flow of protons through proteins in the inner membrane then causes changes in the shape of the proteins in the inner membrane, and they push on other molecules in the ATP synthase. They're pushing on that F1 subunit that changes the shape of it and helps it click together ATP molecules. In the case of the flagellum, the protons are flowing. And when we don't know exactly how this works yet, the molecular details, but it's the MOT-A protein is conducting protons through the membrane the shape of MADE changes and it pushes on some other proteins to help rotate that big base of the flagellum.
Right.
Now there's even more to say. It turns out there's probably proton motive force being used in the secretion part of the flagellum system as well. That was discovered a while ago. Nobody really knows exactly how that works, but bacteria do a lot of surprising things. So often,
And it's just another great example of the cell reusing an idea, this spinning turnstile, and connecting it either to an enzyme that creates things like ATP, or a pump that moves things like proteins, or a hair that rotates the flagellum. It's just interesting that same idea, that same part, is being repurposed in different ways.
yeah, you got to be a little careful. There's
Right.
several different things being used in different ways throughout those systems. But yeah, certainly the proton motive force has been recruited for a lot of different functions. There's a lot of other ones we
Right.
talked about, right? Just pumping toxins in and out, pumping metal ions in and out, right? A lot of those are linked. A proton gradient across the membrane, or sometimes a sodium gradient across the membrane. These are all ways. Basically, anytime you can create differences in concentration, that's an energy source for a cell.
Right. Yeah. So, Nick, the final point then, I want to bring these ideas together. Two seemingly very different stories, the one about the flagellum and the type 3 secretion system, and the one about the ATP synthase. You wrote a paper together with Dr. Mark Palin that explored what I thought was a mind-blowing hypothesis in this paper, and we won't get into the details today. We'll do that next week, but just want to whet our listeners' appetites. You made a number of side-by-side comparisons between these two very different bacterial machines, again, the type 3 secretion system and the ATP synthase. And you described a long list of amazing similarities, not just vague resemblances, like someone who's got a nose and a mouth that makes you think of a certain celebrity, but rather these similarities, there's such a similarity in appearance that you start asking questions and you find out that this might have been a twin separated at birth, or a sibling or a cousin you thought you never knew. So can you just whet our appetite, Nick, for the interview we're going to do next week, where you explore in that paper this idea of the similarities between two very different enzymes, suggesting they share an origin story?
Yeah. Oh, so yeah, I still find it quite amazing myself, right? And the fact having to put together one or two small pieces in that puzzle is quite amazing. I think one way to start is that in the flagellum, in the type 3 secretion system part of the flagellum, there are a series of genes on the bacterial chromosome. And in E. coli, because everything's done in E. coli basically, a bunch of labs got together and numbered or named the proteins alphabetically so that everyone could use the same naming system. And so a bunch of the proteins in E. coli, at least, are together on the chromosome and they're in alphabetical order. And the ones we're thinking about are FlyH, FlyI, and FlyJ. And so in many, many bacteria, many flagellar systems, FlyHIJ are found together in that order on the chromosome. And FlyI is an ATPase protein. And it is what we call homologous. It means it's evolutionarily related. It's statistically significantly similar in the either DNA sequence or the amino acid sequence. It's homologous, evolutionarily related to big protein of the F1 subunit of the ATPase or the ATP synthase.
And remember, these are two completely different proteins for two different machines coded for by two different gene clusters.
And so the FlyI protein and the F1 ATPase protein are quite similar in terms of their structure and even in terms of their assembly. Both of them form a ring of 6 proteins. Both of them either generate or burn ATP. They have the same binding site for ATP. They probably have the same kind of changes in shape when ATP is interacting with them. And so that has been known for a while. And then more recently, we've discovered that FlyH and FlyJ, those other genes that are next to FlyI, they also generate proteins that are very similar to proteins in the ATP synthase. And so there's a little submachine there that looks like it was shared by the common ancestor of those two systems. What exactly it was doing is up for debate, but it was some kind of ATPase activity or ATP synthase activity stuck to the inner membrane. And so it might have been an early version of a secretion system. It might have been an early version of an ATP generation system, or maybe something that could do both. Some systems can sort of go back and forth in both directions of certain reactions. So that's a quite fascinating part of that story. And then, yeah, in some more recent research, we've added a few details to that. So, yeah.
So the experience you just described, Nick, is kind of like walking down the street and bumping into somebody, a complete stranger, but you start to notice similarities in their eye and the nose and the mouth and the chin and everything. And you really start to get the sense this person is somehow related to me. And you find out, in fact, yeah, they had a mother separated at birth, that sort of thing. That's the story we're going to hear about next week.
Right. So the more questions you ask, the more similarities you discover. And you feel like you're really on the trail of something when that happens.
Right.
Yeah.
Okay, so we won't get into the details today. We've covered a lot of ground. Next week, we're going to pick up where we've left off here and explore this idea in a lot more detail. But Nick, I really want to thank you for giving us this backgrounder, not only on the flagellum itself, but also just protein biology and understanding how proteins are just such amazing little machines.
Yeah, sure. And I do encourage, if viewers are having trouble getting this in their heads, right, just go on YouTube and just look up bacterial swimming or flagellum, and you'll see some cool videos that'll give you a pretty good picture of what's happening.
Okay, thanks so much, Nick. I really appreciate it.
Cool. Thank you very much.
Have a good evening.
All right, you too. Take it easy.
Yep. Bye now.
All right, see ya.
You've been listening to Recovering Evangelicals with your hosts, Luke Jansen and Scott Dyer. Send your comments and suggestions for new episodes to lukejjansen@gmail.com. Join our private discussion group on Facebook by searching for the Recovering Evangelicals podcast group. Look for the logo that you always see at your favorite podcast catcher and ask to be let in. Subscribe to lukejjansen.wordpress.com if you want to get advanced releases and group posts, or browse through our archive for a full listing of all our previous episodes. And please help the podcast by sharing with a friend and leaving a review at your favorite podcast catcher.